everybody, a warm greeting from me, Prakriti Neupane. Today I am here to tell about Spirogyra and Paramecium. Spirogyra is a large genus consisting of about 300 species. It is found in fresh water such as pools, ponds, lakes, ditches and slow running water of rivers and streams. Spirogyra are bright, slippery and silky in nature, so are commonly known as pond silk. Structure of Spirogyra. Spirogyra has thallus body. It is elongated, cylindrical, on-branch and silky thread-like structure known as filament. Each filament consists of numerous elongated rectangular cells joined end to end in a row. These cells are separated by a partition wall called septum. Cell structure of Spirogyra. Each cell can be divided into two regions. They are cell wall and protoplast. Cell wall is the outermost protective double layer structure. The inner layer is made of cellulose and the outer layer is made up of pectin. Pectin gets dissolved in water to form mucilage which surrounds the filament and holds mucilage in a sheet. Protoplasm. It contains plasma membrane, cytoplasma, one centrally located large vacuole and chloroplast containing pyronoids. The cytoplasm is distributed in the peripheral region of the cell. The plasma membrane lies inner to the cell wall. It is semi-permeable in nature. A large vacuole is present in the center of the cell. It contains watery fluid bounded by tonoplast. Spirally coiled ribbon-shaped chloroplast containing pyronoids is present in the cytoplasm. The number of chloroplasts ranges from 1 to 16 depending upon the species. Pyronoid is the protein structure present within the chloroplast of certain algae. It helps in the formation and storage of starch. Single prominent nucleus is present at the center of the cell. Reproduction in Spirogyra. Spirogyra shows all three the methods of reproduction. They are vegetative reproduction, sexual reproduction and asexual reproduction. Vegetative reproduction. Spirogyra shows vegetative reproduction in favorable condition. The common vegetative reproduction is fragmentation. Fragmentation may take place due to the mechanical injuries caused by water currents, biting of fishes or by aquatic animals. It may be due to change in pH value, temperature or death of some intercalary cells of the filament. Fragmentation involves breaking down a filament into smaller pieces. Each filament can develop into new Spirogyra by the repeated cell division and growth. Asexual reproduction. During unfavorable conditions, some species of Spirogyra reproduce asexually by the formation of a kinate, a planospore and a zygospore. A kinate. In this method, the protoplast loses water and gets contracted. The contracted protoplast becomes spherical and develops a thick wall around itself due to the extra deposition of cellulose and pectin. This structure is called a kinate. A kinate is the thick-walled, single-cell, non-motile resting spore. It can germinate into new filament under favorable condition. A planospore. In this method, the protoplast loses water and contracts, forming a round structure. It develops a thin wall around itself and forms a planospore. A planospore is a thin wall, non-motile, asexual spore. It can germinate into new filament under favorable condition. A zygospore. At the beginning of the conjugation, the protoplast of the cells forms gamete, but due to some physiological and atmospheric condition, these gametes fail to fuse. They become round and secrete a thick wall around them. They start functioning as asexual spore. They are known as a zygospore. A zygospore is a reproductive cell which is formed without the fusion of male and female gamete. They can germinate into new filament under favorable condition. Now let's talk about sexual reproduction. Sexual reproduction in Spirogyra occurs by conjugation. In conjugation, there occurs fusion of two gametes. In these gametes, one acts as male gamete and the other acts as female gamete. The male gamete passes to the conjugation tube to meet with female gamete. In conjugation, in Spirogyra, all the cells of the filaments can form gamete. They are morphologically similar to each other, so they are also called isogametes. In Spirogyra, two types of conjugation are possible. They are scalariform conjugation and lateral conjugation. During scalariform conjugation, two filaments of opposite strands come closer. They lie parallel to each other. They form a mucilaginous sheath around them. The opposite cells of the filament develop a small outgrowth called papillae. These outgrowth increase in their size and join through their anterior end. The wall between the papillae get dissolved and form a conjugation tube. Now it looks like a ladder-shaped structure due to the conjugation of many cells. At the same time, the protoplast of the cell forms gamete. Among these gamete, male gamete shows motility and passes to the conjugation tube to meet with female gamete. Male and female gamete fuse together to form a structure called zygote. The zygote secretes a thick wall around itself and now it is called zygospore. At the same time, the cells of the another filament remain empty. Lateral conjugation. The lateral conjugation is the type of conjugation that takes place between the adjacent cells of the same filament. As a result of lateral conjugation, the zygospores are formed within the adjacent cells. So lateral conjugation is also called the chain conjugation. Lateral conjugation is of two types. They are direct lateral conjugation and indirect lateral conjugation. In indirect lateral conjugation, the wall between the adjacent cells get dissolved and form a conjugation tube. Simultaneously, the protoplast of the cells make gamete, and after sometimes the male gamete shows the motility and passes to the conjugation tube to meet with the female gamete. Male and female gamete fuse together to form a zygote. The zygote, after the secretion of thick wall around itself, forms zygospore. Direct lateral conjugation. During direct lateral conjugation, the lower cell becomes enlarged and forms a barrel-shaped structure called female gamete, while the upper cell is male gamete, which is somewhat elongated and has pointed end. Here the conjugation tube does not form. The male gamete with pointed end directly persists the septum and forms a hole. The male gamete passes to the hole to meet with the female gamete. Male and female gamete fuse together to form a zygote. The zygote, after the secretion of thick wall around itself, forms zygospore. Now let's talk about Paramecium. Paramecium is unicellular, eukaryotic and aquatic animal. It is found in fresh water such as pools, spawns and slow running streams which is rich in dead and decaying organic matter. It is heterotrophic and holozoic. It feeds on bacteria and protozoa. It shows two types of locomotion. They are swimming and creeping. Food digestion is called cyclosis. Undigested food is digested out through cytophages. Shape and size of Paramecium. It is widely distributed free living ciliate. Its body is unicellular. The size of Paramecium ranges from 80 micrometer to 350 micrometer in length and 170 micrometer to 290 micrometer in diameter. The shape of the cell is like sole of slipper, so it is also known as slipper animalcule. Cell structure of Spirogyra. The structure of Spirogyra can be cell structure of Paramecium. The structure of Paramecium can be studied under two different titles. They are external structure and internal structure. External structure of Paramecium is complex. It consists of pellicle, cilia, oral group, and cytophytes. Pellicle, it is the outermost covering of Paramecium. It is thin, clear, elastic, and flexible. It consists of one kind of gelatinous substance. Like this, it consists of one kind of gelatinous substance, and it consists of cilia in its outer side and trichocyst in its inner side. It provides shape and gives elasticity to the cell. Cilia, the outer layer of the Paramecium is covered with fine hair-like structure called cilia. It arises from the cytoplasma and penetrate pellicle. The cilia help in locomotion and to capture food. Next one is oral group. The oral group is an oblique aperture. It runs backward and is situated at two-third part of the body. The base of the oral group is conical shaped and it is called vestibule. The vestibule connects an opening called cytosome. The cytosome turns into gullet. The gullet is a tube-like structure and called a cytopharynx. The gullet terminates in food vacuole. Cytophage. There is a small opening behind the oral group called cytophage. It acts as anus in Paramecium. Through cytophage, undigested food is passed out from the cell. So now let's talk about the internal structure. Cytoplasma. Just below the pellicle there is cytoplasma. It is clearly differentiated between two parts. They are outer ectoplasma and inner endoplasma. Ectoplasma is thin and endoplasma is granular and thick. Ectoplasma consists of trichocyst and base of cilia. Endoplasma consists of cell organelles and cell inclusions. Trichocysts are spindle-shaped defense organs. They are discharged out as needle-like structures when the Paramecium is stimulated. So trichocysts are also known as defense organs. Next one is nucleus. In endoplasma there are two nuclei. One is larger and kidney shaped. It is called macronucleus or meganucleus. Another is smaller and spherical or round shaped. It is called micronucleus. Micronucleus is present in the cavity of macronucleus. Macronucleus helps in the vegetative reproduction and micronucleus helps in the sexual reproduction. Contractile vacuole. In Spirogyra there are two contractile vacuoles. They consist of 5 to 10 radical canals or feeding canals. They appear as star-like structure. They are present one at each end. The functions of contractile vacuoles are they maintain osmoregulation, they help in removal of carbon dioxide, they help in excretion of excess water. Food vacuoles. In Paramecium, numerous non-contractile food vacuoles are present in the endoplasma. They move by its spreading movement, which is also known as cyclosis. Now let's talk about the life cycle of Paramecium. Paramecium reproduce by both asexual and sexual method. Asexual reproduction is by transverse binary fission and sexual reproduction is by conjugation. Asexual reproduction. A Paramecium cell consists of two nuclei. They are micronucleus, which is smaller, and macronucleus, which is larger. Micronucleus is responsible for reproduction, and when the environmental conditions are favorable, micronucleus undergo mitosis and divides into two, and these two nuclei move to either end of the cell. Similarly, the macronucleus, cytoplasma, and cytoplasmic organelles also get divided into two parts, and at the same time the transverse constriction is made from the both sides. The constriction meet at the center, resulting in two dotted paramecium. Another is sexual reproduction. Sexual reproduction in Paramecium occurs by conjugation. During this process, two paramecium come together side by side. They remain attached to the oral group by some sticky substances, and at the point of attachment, pellicle degenerate and forms a cytoplasmic bridge. The micronucleus also get divided by meiosis cell division, forming 4 haploid nuclei. Out of 4 nuclei, 3 degenerate and only 1 survives. For the surviving nuclei also get divided by mitotically and forms 2 nuclei. Out of these 2 nuclei, one is larger and stationary, it is called female nucleus, and the other is smaller and migratory, it is called male nucleus. The 2 paramecium cells then exchange one haploid micronucleus or male nucleus and separate. The exchanged male nucleus and the stationary female nucleus fuse together to form a diploid nucleus called zygote. The diploid nucleus undergoes mitosis 3 times and gets divided into 8 tiny micronuclei. the original macronucleus disintegrates and the 4 micronuclei expand to form 4 macronuclei. 3 out of 4 micronuclei degenerate, leaving one functional micronucleus. The functional micronucleus divides into two and now the paramecium cells becomes divides into 2 daughter cells, each having 2 macronuclei and 1 micronucleus. Again, the micronucleus divides into 2 along with the division of the cell. Finally, there are 8 daughter paramecium cells, each having 1 macronucleus and 1 micronucleus. Thank you.